Hey everyone, welcome back to Dietitians and Nutrition Support. Today we're going to be speaking about protein digestion and specifically how it relates to us in the field of nutrition support. So protein is a macronutrient, as you're probably well aware, carbs, lipid, and protein. So protein are really large molecules, macronutrients makes sense, and these large molecules are found in the tissues of basically every living thing on the planet. So they are super critical for us as humans to continue living, existing, growing, all those things. They're primarily made up of carbon, hydrogen, oxygen, and nitrogen. Nitrogen is the specifically unique component of protein as compared to lipids or carbohydrates. So we'll get to speak a little bit more about how that relates to us in terms of nitrogen balance, as well as how we're going to be removing that nitrogen so that we're able to metabolize the proteins. So if we are thinking about proteins, they're actually made up of different types of amino acids. There's 20 different amino acids, and we'll speak to which ones those are in just a few moments here. But the thing that makes them specifically unique, that will help to separate what kind of protein or which kind of amino acid rather is which, is going to be the side chain groups. So the basic structure of an amino acid is consistent, as you can see here in our figure, but when you start to switch out the side chains, that's how it's going to differentiate for us which amino acid is which, and which amino acid is which is going to help us to determine what protein we're creating in our chain of amino acids. So let's speak a little bit more about amino acids. There are some essential amino acids, meaning amino acids that we as humans must eat in our diet in order for us to get adequate quantities for us to sustain life. Or there are non-essential amino acids, ones that our body can metabolize in sufficient quantities on its own. We do require all 20 amino acids, so just because non-essential amino acids are made by our body does not mean that they are not important. We definitely still need them for us to be able to function. You can see all of our essential amino acids listed here. Arginine is sometimes considered conditionally essential, meaning depending on the condition of the person, it may or may not be essential. For your average, typical, healthy adult human, arginine is not necessarily considered essential. But for infants, children, or adults who might be experiencing some sort of increased catabolism, critical illness, etc., arginine is considered essential. So what does conditionally essential amino acid mean? Well, that's typically when we see non-essential amino acids becoming conditionally essential for someone based on their disease state, growth state, etc. A good example of that is tyrosine in phenylketonuria. Now, if you're unfamiliar with phenylketonuria or PKU, that is an inherited disorder in which someone is unable to fully break down phenylalanine, and the accumulation of phenylalanine can cause for a whole bunch of other issues in the system. So to try and avoid intake of phenylalanine, if they remove that from their diet or have it in very low quantities, that means that we might need to provide additional amino acids that are synthesized from phenylalanine, which in this case would be tyrosine. So you may be wondering how proteins are made. Well, proteins start out as amino acids. We take our amino acids, our 20 different options, and we're going to string them together and actually bind them with peptide bonds to create, lo and behold, a peptide. So the various types of peptides, you could see dipeptides, tripeptides, oligopeptides, polypeptides, and various different lengths and strings of peptides that can then be turned into proteins. So that's how we go. We go from amino acid to peptide to protein, and then if you consider when we're going to be digesting these, we're of course going to want to go backwards from protein to peptide to amino acid. So it's important for us to note that proteins are genetically determined, meaning we figure out what kind of proteins we're going to make based on what our DNA tells us. We're going to see the DNA be transcribed through transcription into an mRNA or a messenger RNA. And from the mRNA, we'll see that translated to make a protein. So our DNA says make this mRNA, and from the mRNA recipe, it'll tell us which proteins to create. So we already mentioned that proteins are pretty much essential for basically every living organism on the planet. And in humans, they're used for tons of different things. It could be cell growth, development, maintenance, being able to create hormones and enzymes. You may be familiar with some of the proteins that are found in our bloodstream, such as albumin, prealbumin, transferrin. All of those are important things for us to know as dietitians. So let's go ahead and jump to the other screen and talk a little bit about how we are going to get all of our proteins from the food that we eat.
Okay, so let's start talking about protein digestion. If you're a person who's going to eat some protein, obviously that's our little bit of protein here, the first thing that's going to happen is it's going to go into the mouth. In the mouth, you are going to see some little bits of mechanical digestion. Obviously, this person's going to be chewing through their food before they swallow it. But what you're not going to see is a whole bunch of actual digestion or breakdown of the proteins. Really, you're just making smaller bits of food so that it can be swallowed, but not a lot is actually happening to physically break down the proteins that are in the food. So once the food is chewed in the mouth, it goes through the esophagus into the stomach, and when it hits the stomach, that's kind of our first place we're going to start seeing a lot of protein breakdown. So in the stomach, you're probably familiar that the pH of the stomach is about 2. So we know that the stomach is a super acidic place, and this is pretty much because it contains a lot of hydrochloric acid. You'll also see that abbreviated as HCl. Hydrochloric acid is super important because, as we know, proteins, when they're exposed to acids, can start to denature. So this is what we want to happen when we're looking to have proteins be digested. So we're seeing those proteins start to break down because they're exposed to hydrochloric acid. And another thing that hydrochloric acid is going to do is it's going to take what we would consider an inactive enzyme called pepsinogen. And the hydrochloric acid is going to help turn that pepsinogen into what we could consider an active enzyme, which is called pepsin. The reason we don't really want active enzymes to just be hanging out in the stomach, if you think about having active enzymes that can break down proteins, obviously our stomach is made of a lot of proteins, so we don't really want just active enzymes being around willy-nilly in the stomach, just in case we could potentially damage our own tissues. So that's why we're seeing that inactive or zymogen enzyme, pepsinogen, being turned into pepsin when it's exposed to the hydrochloric acid. And this pepsin here is going to help us to take our proteins and it's going to help us break them down to get shorter chains of polypeptides, as well as some amino acids. And remember, our goal is to get to amino acids so that we can absorb them. So one more important thing to note about the stomach is that the production of hydrochloric acid and the release of pepsin is all going to be kind of stimulated and regulated by a hormone called gastrin. Gastrin is created specifically by the G cells that are in the stomach, so it's going to be produced when the stomach starts to have food entering into it. So this gastrin will say, okay, there's food here, now we need to get going, make some hydrochloric acid, and then also start the release of the pepsin once we've seen it become activated from the pepsinogen. So at this point, once we're done with the stomach, you'll see it pass through the stomach and dump into the small intestine. And at this point, we kind of have polypeptides and maybe a few amino acids floating around. While the stomach is definitely a heavy hitter for protein digestion, the actual major site of protein digestion is within the small intestine. So as food comes into the small intestine, we'll start to see the secretion of a hormone called cholecystokinin. It's also called CCK for short. The release of cholecystokinin is stimulated by lipids and proteins coming into the small intestine. And with the release of cholecystokinin, we'll start to see the pancreas becoming a super important player here in our protein digestion. In our other videos, we've talked about pancreatic enzymes And in this particular case, we're interested in those pancreatic enzymes that are breaking down proteins. So we call those proteases. And really, proteases is telling us we're talking about protein, and the ases part is saying it's an enzyme. So any protein breakdown enzyme is going to be considered a protease. So our three primary proteases that are working in the small intestine that are produced by the pancreas are trypsin, chymotrypsin, and carboxypeptidase. All three of these different proteases work somewhat similarly, but they're going to focus on cleaving specific types of bonds that are seen within our polypeptides to break them down all the way to our amino acids. So technically, once we've seen our proteins get broken down, we'll have kind of a mix of primarily amino acids, as well as a few dipeptides and tripeptides. So after we've done all of our protein breakdown through the stomach and the small intestine, then we're going to start to see absorption of that mix of amino acids, dipeptides, and tripeptides. This is going to take place in the small intestine, and it's going to be a mix of active transport and facilitated diffusion. This either means that with the active transport, it's going to require ATP, which is kind of a type of cellular currency, or with the facilitated diffusion, that means that we're going to see some type of a transport protein that's going to be required to be able to help move the proteins that we want to absorb from the outside of the cell into the inside of the cell. This could be a sodium and potassium pump, for example. So once we see all of our proteins that have made it through all the loops of our small intestine, we've absorbed what we want to of our amino acids, dipeptides, and tripeptides, and for the most part, we have really good absorption of all of those. Once they're absorbed into the cells of the small intestine, or enterocytes, as you might also hear them called, you'll see that the proteins, or these amino acid pieces of the proteins, are going to move directly into the bloodstream. Once they're in the bloodstream, they've really entered into our amino acid pool of sorts in the body, because remember, there's no actual reserve storage place for amino acids, so we really just have amino acids kind of moving and flowing throughout the bloodstream. We're going to see all of our amino acids transported to the liver, and this is going up through that hepatic portal vein, and this is going to allow the liver to make the decisions on where to distribute those amino acids into the various places that might need them throughout the body. So once they've gone from the liver all to the various different parts of the body and are utilized, we'll see the continuous turnover of proteins throughout the system. So if we use our proteins to build some cells, those cells are remodeling themselves, we're going to see buildup and breakdown consistently happening from all of these different proteins and amino acids. Now, because proteins and amino acids are nitrogen-containing compounds, once we've seen all of the breakdown start to occur of the proteins and amino acids that we've consumed, we start to see this nitrogen need to get removed from the body. We'll actually see the nitrogen in the form of ammonia. And if you think about it, should you really have ammonia just go and loosen the body? No, so our body's very smart and it will package the ammonia up in the form of urea. This whole conversion process happens in the liver as well. Thankfully, urea can actually be excreted by our kidneys, so we'll see that urea going to the kidneys and being urinated out. If you're digesting the protein appropriately, you shouldn't really see a lot of protein getting into the large intestine. You should have really broken much of it down and absorbed pretty much all of it in the small intestine. There are some cases if you consume very, very extreme amounts of protein that you might see some get into the large intestine. It's not really a great thing. The microbes in your small intestine will go to town on the protein, and this can cause you some GI distress and could cause you some other GI issues if that protein starts to ferment. And as we already kind of mentioned, we have this pool of amino acids that is going throughout the system. And so really we have kind of a balanced level of how many we get to keep in this pool. We're trying to provide those amino acids to where they're needed in the system. And at some point, we might have a little bit of extra protein. You should not see a lot of protein that's going to be leaving the system through the kidneys. Protein is a really large molecule, so if we push the protein molecules through the nephrons and through the glomerulus within the kidneys, which are very, very small, we could really see damage actually happening to the kidneys. So you only really are going to see protein excretion in an excessive amount if the kidneys are already damaged and it's pushing that protein through them. So we don't want to see a lot of urination of protein, but if you have excess protein that your body is not really using, you will see that start to be stored. You'll see the amino acids that cannot be used be converted into lipids, and then they'll store in usual lipid storage locations, those adipocytes, and various fat depots. So there's our short walkthrough of protein digestion. Let's jump back to our other screen so we can talk more about the details of how much protein you might need in your diet.
So now we know a little bit more about how protein is digested and absorbed into our system, but let's think again about protein in the diet. When we're thinking about protein in the diet, we're typically going to be consuming all 20 amino acids in the variety of foods that we're going to eat throughout the day. But what we're going to think about in terms of protein-containing foods, and specifically complete protein foods, those are going to be the foods that contain all of our essential amino acids. And not only do they contain all of the essential amino acids, but they have them in the appropriate balance that is most appropriate for stimulating growth and development and all those things in our body. For protein-containing foods, we can see something called a limiting amino acid, and this is going to be referring to a specific essential amino acid that may be at too low of a quantity or might be missing from a food so that it's keeping the food from being considered a complete protein. While we might consume a variety of incomplete proteins as well as complete proteins in our diet, you can see the use of complementary foods or pairing multiple foods together to get all of the essential amino acids in our diet in an adequate level. When we're considering how much protein is enough protein, the RDA, or the recommended dietary allowance for adults, is going to be 0.8 grams of protein per kilogram of actual body weight. This is just enough protein to meet the basic requirements for that average adult person. This is not going to be enough for someone who is doing intense exercise, who is critically ill, or who might have some kind of a different disease state. We're going to see different recommendations for them, but just that basic nutrition recommendation is going to be 0.8 grams per kilo. The acceptable macronutrient distribution range for protein is also somewhat wide. It's 10 to 35% of each day's total calories allotted to protein. On the high end of things, we typically see a maximum recommendation of 2 grams of protein per kilogram of actual body weight. This level of protein intake is considered safe without adding additional burden on the kidneys for the need to excrete extra nitrogen. For people who are consuming protein at this high level, we typically recommend an increase in fluid consumption so that they can help to have better GI functioning, as well as that they'll have more fluids in their system to be able to flush out and excrete the additional urea that's coming from all of the nitrogen groups on the proteins. And speaking of nitrogen, we can also consider nitrogen balance, which is going to tell us a little bit of information about whether or not our body is at kind of a homeostatic level of protein in the system, if there's perchance growth or development, or if we are having inadequate intakes of protein and we're actually breaking down and losing more protein than we are intaking. For the most part, your healthy adult person is going to be in nitrogen balance. They're going to have a approximately equal quantity of intake and output each day. You'll also see positive nitrogen balance, in which the nitrogen consumption is larger than the quantity of nitrogen excretion. This is going to be present for infants and babies and growing children, as well as for individuals who are pregnant. On the opposite side of things, you can see a negative nitrogen balance in which someone is outputting more nitrogen than they are consuming. This is commonly seen in malnutrition or low protein and low calorie diet intakes. It can also commonly be seen during severe illness, infection, following burns, and other critical illness states. For patients receiving clinical care, there is a way for us to estimate the quantity of nitrogen output as well as nitrogen input to assess nitrogen balance. The first step of determining nitrogen balance is going to be figuring out what the nitrogen output was. Determining nitrogen output is going to be a two-part piece of first determining the urinary urea nitrogen output that you're going to see from the urine across a 24-hour span, and then we'll add an estimated ballpark for the quantity of nitrogen that we presume the person is going to lose through non-urinary sources. The next thing we need to do is determine the nitrogen intake. We can do this by taking the quantity of protein that someone consumes during the day from counting up all the grams of protein within their diet and then dividing that by 6.25. This will get you the estimated nitrogen intake per day. And then finally, we can get the nitrogen balance by taking the nitrogen intake and subtracting the nitrogen output from urinary and non-urinary sources. If you're in nitrogen balance, the value will be 0. If you're in a positive nitrogen balance, then you'll see a positive value, versus if you're in a negative nitrogen balance, you'll see a negative value. So let's think a little bit more about how protein is going to play a role in our nutrition support provisions. From an enteral perspective, our standard formulas are also going to be considered polymeric formulas. This means that they have whole intact proteins within them. For the most part, all of our formulas, unless they're for a specific disease state such as phenylketonuria, like we spoke about earlier, are going to contain all of our essential amino acids. They're all going to be in that whole intact form, and that's going to anticipate that the person consuming them is going to be able to break down the whole proteins all the way down to the amino acids and peptides for absorption. Next, you'll see semi-elemental formulas. These formulas have slightly broken down proteins. You'll see the proteins in the form of oligopeptides, meaning they're just broken down a little bit. They're not all amino acids, so it's semi-elemental. And finally, you'll see formulas that are considered hydrolyzed, monomeric, or elemental formulas. This means that they have had their protein completely broken down and it is going to be amino acids within the formula. This is going to be really helpful for patients, specifically those who are being fed lower in the GI. So if you have a tube feed that's going to the small intestine, for example, we're bypassing a lot of the digestion that happens in the stomach. So we'll want to make sure that we have a partially broken down or fully broken down formula so that that person is able to tolerate it. This can also be helpful for patients who are experiencing GI dysfunction or who we may have some concerns about their ability to break down protein fully. And for the most part, we're not going to start someone on an elemental formula unless they're showing intolerance on a more intact protein formula. Now, from a parenteral nutrition perspective, we're going to see our parenteral nutrition also containing all 9 essential amino acids. We'll see the amino acid concentration ranging anywhere from 3.5% to 20% for our amino acid solutions. One lab value for you to be specifically paying attention to in terms of protein intake is going to be the blood urea nitrogen, or the BUN value. We want to look at this closely because we can sometimes see people experiencing azotemia, which is a BUN greater than 100 milligrams per deciliter. So we already know that protein is going to be that main source of nitrogen in the body, and we're going to need to excrete that as urea. If you have excess levels of blood urea nitrogen, that means that you have more urea within your system than you are able to excrete currently. And so having an increase of protein or excessive intake of protein is really just going to keep adding more urea that your kidneys at this point are not showing the ability to excrete efficiently. Sometimes this can occur in critical illness and sometimes in older adults, but we really want to be mindful of kidney function and not end up having too much excessive urea within the system. So specifically for those individuals that you're starting parenteral nutrition on, if they are experiencing azotemia, you really want to be mindful and very cautious in the way that you approach your parenteral nutrition, as not to cause them to have further concerns with elevated urea nitrogen in the bloodstream. We've gone through a lot of material today talking about protein digestion and absorption, as well as all the different ways that it might impact our nutrition support practices. If you have any questions about protein, please feel free to leave them in the comments boxes below, and let us know if you have any ideas for future videos as well. Thank you so much. We hope this was helpful, and we look forward to seeing you next time.